In this video, we're going to be looking at incomplete dominance. So under ordinary situations, you would have one allele that's dominant to another allele, which is recessive. In the case of incomplete dominance, neither allele is actually dominant to the other. So what that means is that if both alleles are present, they're going to have a complex interaction together. So we're going to work an example problem for this. And under ordinary situations, we would use a capital letter to represent a dominant allele, and we would use a lowercase letter to represent a recessive allele. Since neither allele is dominant in this case, I can't do that. So instead, I'm just going to choose a letter to represent the characteristic. So let's say in my example, I am looking at flower color in snapdragons. So for this allele, I am going to use a capital F to represent flower color. And there are two different alleles for flower color. There is one allele that is red, and there's one allele that's white. Both of these alleles show incomplete dominance. So what that means is that if I'm going to cross them, I need to use the capital F to represent my flower color, and then I'm going to put a little r up top to represent the red flower allele. I have to do it like this up top because I can't use the capital and lowercase like we normally do. So if I have a flower that is red, that means that it has two of the red alleles. So I will write this FR, FR. And I'm going to cross this with a flower that is white. So in a white flower, I would write this using a W to represent the white allele. So this would be FW, FW. I'm going to set up my cross with a Punnett square, exactly like I would for a simple dominance cross. And I'm going to put one parent on the left. So this parent could either give an FR, or they could give this other FR. And my other parent on the top. This parent could give an FW, or they could give their other FW. Now in terms of putting things in the boxes, normally we put the dominant allele first and we put the recessive allele second. But in this particular case, nothing is dominant, nothing is recessive. So instead, we just keep everything alphabetical because that's going to make it simpler to keep track of. So I have an FR from here, and I have an FW from there. Same thing here, an FR, and an FW from the top, an FR from here, and an FW from the top, and my final box is going to look exactly the same. FR from the left, FW from the top. When I'm writing out my genotypic ratios, in the past we always listed the dominant first. But in this case, nothing is dominant. So once again, I'm going to follow the rule of keeping it alphabetical. So my first possible phenotype is going, or genotype rather, is going to be FR, FR, followed by my FR, FW, and last of all, my FW, FW. So you can see that I've grouped these where all of the R's are first, all of the W's are second because they are alphabetically later. So counting them up, I have 0 that are FR, FR, 4 that are FR, FW, and 0 that are FW, FW. For the percentages on these, that would be 0%, 100%, 0%. My phenotypic ratio is going to be a little bit different. So if I have two red alleles, that plant is going to turn out with red flowers, just like this parent one. So I'll list red first. And I'm listing my phenotypes in the exact same order that they appear in my genotypes. I am not continuing to make this alphabetical. Now, having one red allele and one white allele, since these are incomplete dominants, that means these guys are going to mix. Red and white, when they mix, is going to give me a pink. So these flowers that have one of each allele, they're going to turn out pink. My last one is my FWFW, and that would be white. So again, I count them up, 0, 4, 0, and that is 0% red, 100% of these offspring pink, 0% of the offspring white. And so typically what's going on in this sort of case is that both of these alleles are coding for making a particular protein. In this case, it's coding for making a protein that gives it a reddish color. If it has 2 copies of that allele, it's going to make lots of the reddish protein and will turn out red. If it has no copies for that allele, it makes none of the reddish protein and it appears white. Whereas if it has 1 copy of the red allele, it's going to make some of that particular protein. And that's why we are getting this thing that is somewhat in the middle. So again, in terms of working the crosses, this is going to be done exactly the same way you would have done your simple dominants, except instead of using capital and lowercase, we're using one letter to represent the trait and another letter up top that helps us keep the 2 different alleles separate. So a R up here to represent my red, a W to represent my white. The other thing to notice is that if you've done this correctly, your genotypic and phenotypic ratio are going to be identical when you are doing problems that show incomplete dominance.